I'd like to call Jason Brzezinski to the stand. Could you state your name for the court, please?
Jason Brzezinski.
What is your occupation?
I'm a forensic DNA analyst for the shoe crime.
How long have you been employed by the shoe crime lab?
I've been employed for 1 year.
What is your educational background?
I received a Bachelor of Science in Forensic Science from Seton Hill University, where I completed coursework relevant to DNA analysis, such as genetics, forensic molecular biology, and statistics.
What are your specific duties and responsibilities as a forensic scientist in the DNA analysis unit?
My duties include extracting DNA from evidence that we get in the lab and documenting those procedures in notes, detailed notes. I also apply statistics to samples of DNA once they've been amplified and quantified.
Have you received any specialized training in the field of DNA analysis?
Yes, I have, and I've also completed a proficiency test given to me by the shoe.
Have you conducted any research in the field?
Yes, I conducted a research study my senior year at Seton Hill University.
Are you familiar with the published journals of forensic DNA analysis?
Yeah, I believe it's important to keep up to date on the rapidly evolving field of DNA analysis.
Do you belong to any professional societies?
No, I don't.
And have you testified in court before?
No, I haven't.
I have no further questions. And I move to qualify Jason as an expert.
Do you have any questions? Yes, I do, Your Honor. Dr. Brzezinski, is it?
Yes.
Where did you get your doctorate degree?
I did not get a doctorate degree. Bachelor of Science from Seton Hill University.
And do you have a master's degree?
No, I do not.
Are you aware of master's programs in forensic science?
Yes, I am.
Isn't it true that if you had a master's degree in forensic DNA analysis that you would be better qualified to
No.
perform the analysis?
No, not necessarily. We receive all the relevant training at Seton Hill University, and then training from the lab, and our proficiency test verifies that we have the proper knowledge to address our duties at the Seton Hill Crime Lab.
And what is your bachelor's degree in?
Forensic Science.
So it's not in forensic DNA analysis?
No.
So shouldn't you have at least one degree in forensic DNA analysis in order to be considered an expert?
No, like I said, we complete courses relevant to DNA analysis during the course of that, as well as courses that address other forensic procedures. And again, like I said, we have training at the lab, and we do a proficiency test to make sure that we're up to the national standards on DNA analysis.
So what is this proficiency test?
It's a test that's given to us by the lab. What happens is a third party administers this test so that there's no tampering or anything from our lab. And they send us usually a case that involves DNA, and it's blind, so we don't know that it's even real or a test. We would process it like any other test. We would get our results, and then we'd send them back. And that third party that administered the proficiency test would then verify whether or not I was correct in my analysis of the DNA.
And so you've completed these tests.
Yes.
Have you passed these tests?
Yes.
So you've gotten the correct result verified by a third party.
Yes.
Have you ever been qualified as an expert before?
No.
So you don't meet the qualifications to be an expert in DNA analysis?
I do.
But you just said you weren't qualified as an expert.
This is my first time being qualified as an expert in the court.
So you're not qualified?
Not until we complete this qualification.
And so you're not qualified as a forensic DNA analyst. Are you familiar with something called certification?
Yes.
And could you please explain what that is?
Certification is a standard that's used to verify that an individual is certified to analyze DNA in the lab and that they're following the proper procedures and protocols given by that lab or by the state, whatever the protocol calls for. And that certifies, it's usually like a certificate or some sort of merit that you've completed your proficiency tests and any other relevant work that would allow you to analyze DNA in the lab.
Okay. And are you certified?
Yes.
Say no. May I approach the witness, Your Honor?
Yes.
Could you read This is from NPR. Are you familiar with the organization NPR? It's National Public Radio. And it's a reliable source of information. Do you agree?
No.
No?
I think that it depends on who's providing the information to NPR, whether or not it's verified through scientific methods or studies or groups. If it was medical, verified through the CDC.
Well, we're going to look at something that they have stated. Would you mind reading this paragraph here where I have a little check mark from here to here, please?
Yes. With no central governing body and a lack of mandatory standards and certification processes, the practice of forensics is unregulated on a nationwide level and highly uneven. As a result, there is wide variability in forensic methodologies, reliability, and types and numbers of potential errors.
Okay, so the NPR, questionable if you believe that they're reliable or not, but they're saying that the certification isn't really governed within crime labs, and it does lead to potential errors.
I would disagree. The ASCLD lab actually provides a national standard that all labs adhere to, and they can administer certifications and verification that the labs and the individuals themselves are following proper protocol and procedures.
So ASCLD provides certification? ASCLD lab provides certification?
They provide, yes, certification.
What if I told you that they don't provide certification? They provide something called accreditation.
Yes, they accredit the labs.
Yes. Certification will be provided by a different agency. So we'll say that you are certified. Okay. So, but we don't necessarily know that this is a good thing because it's not regulated according to the National Public Radio. Are you familiar with quality control and quality assurance?
Yes.
Yes. Could you explain to the court what those things are?
There are measures that are taken within the lab to make sure that the processes that we do in the lab are adhering to the protocols set forth by the lab or any governing body. And they can vary. You know, there could be check-off sheets and sign-off sheets similar to a chain of custody. It depends on what process you're doing, but for instance, if you were taking something out, you would want to sign it out, and then when you're doing analysis, there may be protocols on how much of each reagent to add, and you would sign off on a sheet to make sure that you're doing all of those protocols properly, and then there's someone in charge of maintaining those records and making sure that everyone's following protocols.
Okay. So this would be considered a good thing.
Yes.
And do you follow quality control and quality assurance guidelines?
Yes.
You didn't testify to that yet? No. So how do we know that you follow these guidelines?
I take proper notes when I'm doing my DNA analysis, and the labs provide the quality assurance, the quality control, and quality assurance.
Your Honor, I object to Mr. Bishevsky being entered as an expert. He lacks any degree in forensic DNA analysis, and his certification is questionable.
What is DNA?
DNA is a genetic blueprint that gives us our physical attributes and other characteristics.
And where is this DNA found in the human body?
DNA is found in every cell in the body except for red blood cells, and this is due to a lack of a nucleus within the red blood cells.
Does DNA vary from person to person?
Yes, it does, except for in identical twins.
Can you identify these differences?
Yeah, we look at 16 different locations within someone's DNA, and these are all known to be highly variable between person to person.
Is it possible to perform DNA analysis on a dried body fluid stain?
Yeah, several of the stains that we analyze in the lab come from a dried stain, which we extract from, and the dried stain is actually a good way to store it. If you store it like a plastic bag, or it's not dried, it can form moisture, and moisture can degrade the sample.
And what is the procedure you use for examining DNA?
So to examine DNA, we get samples in the lab. We get known samples, which are swabs or blood draws from a person who we knew who we took it from. We also get unknown samples, which haven't had their origin determined, or the person that they could have come from determined. We take a small portion of the question sample. We add chemicals to it that break apart the cells which house the DNA. Then we use another chemical that kind of washes away all that extra material, extracellular material that we don't need for analysis. We copy the DNA, we make sure that we have enough DNA to work with, we copy it, then we analyze it. Now, if even one location varies within that DNA analysis, we can exclude the known sample from contributing to that unknown sample. If they match at every location, then we would run further analysis, like statistical analysis, to give us more details on how likely it is that somebody else could have provided that.
Is this technique performed only at your crime lab?
No, it's done at crime labs worldwide.
And is this a new technology?
No.
How do you know that you're getting the correct result?
What we do is, the first thing we do when we get evidence in the lab is we check it. We check the packaging, we check for signatures, dates, the chain of custody, just to make sure that it hasn't been tampered with. We want to make sure of that first. And then after that, we make sure that all of our work areas are wiped down with bleach, and we continue to do that through. We make sure that our work area is clean throughout. We don't want to mix up samples between the knowns and the unknowns, so one of the things that we do is we process our questions before our known samples. What this does is it prevents any contact between the known sample and the unknown sample, and any exchange of biological material that could tamper with or falsify the results of the unknown. And then throughout, we use reagents throughout our analysis to make sure that all the procedures that we're using are being run correctly. Reagent blanks make sure that our extractions are pure, and positives make sure that the instruments are running correctly, and then we run again negative, which lets us know that our reagents are pure.
No further questions? Did you have the opportunity to analyze evidence in case number 4 involving a victim by the name of Ashton Butcher?
Yes, I did.
And what evidence did you receive?
I received a cigarette butt from the origin of fire at the Butcher residence, a known blood sample from the victim, Ashton Butcher, a known blood sample from Demi Les, and a known blood sample from Charlie Sheets.
What did you do with the evidence once you received it for analysis?
First thing I did was, like I had mentioned before, I checked the packaging to make sure there was no tampering, dates and everything were in order on the packaging and matched up with the chain of custody. Then I extracted DNA from the cigarette butt. I extracted from 2 locations, the end of the cigarette butt, which typically would be in the mouth, and then a location that appeared to have lipstick. And then I compared that extraction of that DNA to the known results from all 3, Charlie Sheets, Demi Les, and Ashton Butcher.
Did you analyze the evidence using the techniques you described earlier?
Yes, I did.
May I have permission to approach the witness? Yes. Do you recognize this document?
Yes, I did.
Could you please tell the court what the document is?
Yes, this is a report of my examination. I prepared this myself, and it gives the results of my analysis on the DNA samples.
Is that your signature at the bottom?
Yes.
Has the copy been altered in any way?
No.
Thank you. I move to enter the report as states exhibit number 1. Did you compare the DNA from the cigarette butt from the origin of the fire at Ashton Butcher residence to the known blood sample from Demi Les?
Yes, I did.
Did you include in the report the results of your analysis on DNA from the cigarette butt?
Yes, I did.
And what were these results?
The results matched from the cigarette butt and the known sample from Demi Les.
Did you also compare the DNA from the cigarette butt from the origin of the fire at the Butcher residence to the known blood sample from Ashton Butcher and Charlie Sheets?
Yes, I did.
And what were the results of that comparison?
Those results did not match either of the known samples from Charlie Sheets or Ashton Butcher.
So can you say with some degree of scientific certainty that the DNA from the cigarette butt from the origin of the fire at the Butcher residence is not from Ashton Butcher and Charlie Sheets.
Correct, it is not.
And can you say with some degree of scientific certainty that the DNA from the cigarette butt from the origin of the fire at the Butcher residence is from the accused Nemulas?
Yes, it is. After I analyzed the DNA from the cigarette butt, I compared it to the known sample from Nemulas, and I applied statistical analysis to it after all the matches were made, and the statistical analysis revealed that the chance of finding another random, unrelated individual in a Caucasian population would be 1 in 165 quadrillion. That's 165 followed by 15 zeros.
Thank you. I have no more questions.
I have some questions, Your Honor. Dr. Bishevsky, where is DNA found?
DNA is found in the cells, in every cell in the body except for red blood cells.
So, if isn't found in red blood cells, what were the samples that you got from the victim and the suspect in the case?
Those were known blood samples.
How would you get blood from the known blood samples if there isn't DNA in red blood cells?
There is not DNA in red blood cells, however, blood is not entirely comprised of red blood cells. There's also white blood cells and platelets.
So, where did you get the DNA from then?
It could have been the white blood cells from that blood sample.
Okay. Well, what about then the cigarette butt? Where did you get the DNA from the cigarette butt?
More than likely, it could have been saliva from the mouth. Had it been in somebody's mouth, it could have been from the hands touching it, sweat. So again, saliva from the mouth, sweat from the hands. At one point, the cigarette it appeared to be smoked down. So I'm assuming at some point it was held and more than likely put in somebody's mouth to smoke.
Does saliva have DNA?
Yes.
Saliva has DNA in it?
Yes.
I have reliable sources told me saliva does not have DNA.
Saliva does have DNA.
Are you certain?
Yes.
It's not the cells, skin cells that are found in saliva?
So, yes, the cells inside of saliva are what contain the DNA.
Alright. How big is DNA?
As far as
Molecularly, how big is the molecule of DNA? Not specifically, but just in generalities.
it's very large.
Very large.
Yes.
And did you say that
I
you
mean
did
relative.
something-
Very large relative to how small
Okay.
it is.
Did you testify to something called PCR?
I'm sorry?
PCR?
Yes.
Could you explain again?
Preliminary chain reaction.
Yes, what is that?
It's a way of amplifying and analyzing DNA.
Okay. So what does this mean, amplifying?
Copying the DNA.
Okay. So you copy the DNA in order to test it.
Yes. I mean, the whole testing procedure, depending on your definition of testing, I guess, the whole test itself, there would be extraction, quantification, then we would amplify it, and then within that amplification stage, it's put into a thermocycler. And now there's different stages within that thermocycler as well that allow for the DNA to be copied. And then we can analyze using an instrument as well to compare the DNA to known samples.
So it's just a portion of the analysis, this PCR.
Correct.
Is that what you're saying? Okay. Now you said you made copies. Do you copy that whole huge molecule of DNA?
We copy target regions of the DNA. What we do is we heat up the DNA, and whether you were DNA is double-stranded. What happens when you heat it up is it separates, it denatures, so those two strands become unwound. We then put in primers that are targeted towards those target regions, and we anneal the DNA, and that allows the primer to adhere to the DNA, and it begins to copy it, and then it'll cut off at the target region that we're looking for. And depending on which kit you use, there's different target regions.
So how many target regions do you look at?
We look at 16 different target regions.
Okay. So DNA is huge, but you only look at 16 areas?
Well, there's variance in the DNA, in the complete chain of DNA. So even within that small area, there could be tons of differences as far as the order of nucleotides and.
But doesn't it make sense that if you would have looked at more areas of the DNA, that your test would be more reliable?
No. Once you get a result, well, if you get a result from your analysis, that result is steadfast. If you get a result, you get a result. More DNA doesn't necessarily mean better results or the likelihood of results. You can run your analysis on the amount of DNA. PCR takes very little DNA to use compared to some of the other methods that have been used in the past, and that's one of the benefits of it. That's why we use it.
But you said that you looked at 16 areas?
Yes.
Is that right? Wouldn't it have been better to look at 20?
No.
No?
Those 16 regions are what has been the standard, and that's what we use to compare to every other sample.
May I approach the witness, Your Honor? Yes. Is this also an accurate copy of your report?
Accurate except for these highlight marks.
Okay, but mine is the highlights. I did the highlight. Okay. So, I have highlighted some areas. Item number 1A. What's item number 1A?
Item number 1A is going to be cutting from the butt of the cigarette. This will be items and marks.
Okay. And what is item number K1?
K1 is a known blood sample from Ashton Butcher.
And that's the victim who resided where the cigarette butt was collected.
Correct.
Is that correct? Okay. So how do you go about comparing the DNA? You look at these 16 areas and look for similarities between the question sample and the known sample. Is that correct? Okay. So what I've done is I've gone down through our question sample and our known sample, and I found numerous areas of similarities between the cigarette butt and the victim, Mr. Butcher's DNA. And to me, it seems like their DNA is very similar, and I'm wondering why he isn't included as a source of that DNA.
Well, if you look, these are going to be your allele pairings found within that DNA. So, at each location that we look, there are certain allele pairings that we can have. Now, they both have to match for it to have come from the same person. If there's a difference, it's going to exclude that person. And kind of one of the ways you can look at it is here at the amelogement. This is what we use to type the sex of the individual. So we have an XX, which is a female. Then we have an XY, which is a male. So obviously, just because there's an X and an X wouldn't provide for the same individual.
So the fact that he has an X, and we see an X in the question sample, doesn't include him as a source?
It's the combination of the alleles that's important. So, for instance, you're going to see here a 9, 9, and then a 9, and a 9.3. That's going to exclude at that location the sample from contributing to the known. And once again, like I said, if you look at the amelogement, it's important to look at the pairings. You would need 2 X's for a female and an XY for a male. So it's really the pairing that's important.
Okay, so you have to have both.
Correct.
In order for it to be considered.
Correct.
Okay. All right. Can we refer to your chain of custody sheet, please? And your report. So I noticed on your report you have an item 1A and an item 1B. Is that correct?
Yes.
But if I look at the chain of custody sheet for the items that you received, I only see an item 1. I do not see an item 1A or an item 1B.
Correct. Item 1 was received as a complete cigarette butt from the origin of fire at the Butcher residence. Upon receiving it and inspecting it for tampering, I opened it and I wanted to extract from locations where there possibly could have been DNA left behind. So I extracted from what I refer to as the end of the cigarette, the part that would have been in the mouth. And like I said, looking at the cigarette, it appeared that there was a pink smudge mark a little bit higher up on the butt of the cigarette. And I wanted to extract from there as well. Assuming that it was lipstick, my assumption was that there might have been DNA left behind that would have provided another piece of analysis for me to look at. So on the report, item number 1 is what I received, and then I detailed 1A and 1B as what I did to that evidence thereafter within the lab.
I see. So that is actually part of what item number 1 was.
Correct. Both 1A and 1B were part of number 1.
Now, what date did you receive these items of evidence for analysis?
I received them on October 29th, 2013.
And do you know when they were collected for analysis?
When they were collected for analysis.
When the crime occurred, right, yes, when they were collected.
They were collected on 9/28/13.
So you didn't receive them for over a month after
Correct.
they were collected?
Correct.
Isn't it true that those samples could have degraded in that amount of time so that you would have gotten a false match to my client, Ms. Debbie Less?
It depends how they were stored by the holding agency that would have collected them. Sarah Siddle, I believe. Whoever she submitted them to first. It would depend on how they were stored from that agency, but upon my receiving them, they were stored in a freezer, and that would have prevented any degradation from thereafter.
Well, do you know how they were stored from the time that they were collected until they came into your possession?
I do not.
But it is true that the samples could have degraded, that you got a match to Miss Less when you shouldn't have gotten a match. Is that correct?
No. Had they degraded, we wouldn't have gotten results.
What do you mean by not gotten results?
We wouldn't have had the fact that we got results, so the fact that we got any reading at all from the DNA through my analysis and through the instrument, the gene mapper instrument, or ID software, had they degraded, we would have seen that in the results. We wouldn't have had this complete of a profile. And as you can see, item 1B, there were no results at certain alleles.
So you're saying that you would have gotten results, they would not have falsely matched my client?
No, we wouldn't have gotten results.
Well, I'm glad that you brought up this where no results were called, because item 1B is still part of the cigarette, is that correct?
Correct.
And I do see the stash and it says no. Why are there no results called? Is that because they didn't match and they could have excluded my client as a source?
No, what could have happened is item 1B was where the lipstick stain was, further up on the cigarette. So, like I said, it was my assumption that if it was lipstick, it had touched part of the mouth. It may have been that that portion of the cigarette had either little to no DNA actually applied to it, because again, not being there, that was just an assumption I made. Or that specific part of the cigarette could have been degraded. More importantly, the 1A, which is from the same cigarette, same piece of evidence, did provide a full profile.
Now, where was the cigarette butt collected from?
It's collected from the origin of fire in Butcher residence.
Can you say that cigarette butt was used to start the fire?
I cannot.
And can you say how that cigarette butt got there?
I cannot.
So you can't say that cigarette butt was, I guess, deposited at the crime scene in the commission of any kind of crime.
I cannot, other than I can say that the cigarette did at some point come in contact with Miss Debbie Less.
So can you say that she smoked the cigarette?
No, like I said, it could have been her hand that touched the end of the cigarette, more than likely because of the location of the cigarette. The general assumption would be that it touched the mouth at some point, or the hand, but regardless, there was DNA left behind, and we did get a complete profile from the from the unknown cigarette butt that we could compare to Debbie Less's known blood sample.
So is it true then that you're saying from her touching it, could she have just pulled that cigarette out of a pack of cigarettes and handed it to somebody else to smoke? And that's how you got her DNA on it? That she just gave it to somebody else just by touching it?
Theoretically, yes, she could have. That cigarette would have had, more than likely, had DNA from one of the other individuals, if those individuals were involved at this scene. So assuming, with what I have known, again, we have a profile that matches Demi Les from the cigarette butt.
But her DNA could have gotten there by just handling the cigarette?
Yes.
Okay, she didn't have to smoke it?
Correct.
Okay. Somebody else could have smoked it?
Correct.
Okay. So there really is nothing tying my client to the crime scene?
Her DNA was found on the cigarette at the crime scene, so she can be tied to the crime scene in that sense that, again, her DNA was found on the cigarette, and I made that match with the known and the unknown.
How much DNA did you get off of the cigarette butt?
How much, like, of an area did I extract from the cigarette?
No, how much DNA? Like, if you could quantitate it, how much was present?
Do you want to know from both of my cigarette cuttings?
Yes.
Okay. From the area with the supposed lipstick, it would have been 0.0787, and from the end of cigarette, 0.526.
So what does that mean, 0.0787?
It would have been
So, is that a lot of DNA or is that a little bit of DNA?
That's enough DNA to run through a PCR analysis and get a full profile, as you see here. Again, very little is needed.
So very little is needed.
Mm-hmm.
Isn't it true then that very little is needed to contaminate that sample?
Had there been contamination, yes, but we would have seen it after my analysis.
So
We would have had, this would have matched up exactly with Demi Les. Had we had any differences, we could have excluded any other samples.
how do you know that the DNA from Miss Les wasn't accidentally contaminated into the cigarette butt, and that's how you got her DNA there?
By accidentally contaminated.
By you putting it there?
By me putting it there.
Yes.
I've had no contact with Demi Les, and like I said, we processed the question sample before we ever even come in contact with the known sample. And they're in tubes that are labeled, properly labeled, in a whole different work area from where we process our known samples.
So there's no possibility that you contaminated that sample or mixed up this sample from the cigarette butt with my client's sample?
There's no possibility that it happened here. There are possibilities, but that's why we follow the protocol of labeling, cleaning, wearing gloves, working at different areas where we process the question and known. However, being that I follow the protocol, there's no possibility.
Just one more line of questioning, Your Honor. Can we refer to the 310 sample grid, please? Can you please explain to the court what this is?
Yeah, so we have different samples being run through the instrument in the lab. What this is a sign-off sheet showing exactly where on that grid. We have a grid of tubes, essentially. This shows exactly what was put in the tubes and by who, in reference to their location on that grid.
Okay. So, what is this to tell the instrument where to go to look for the sample, or what's the purpose of it?
The instrument is programmed to know where to draw.
How do you know that the instrument didn't go into the wrong tube?
There is quality control methods that are applied to the machine, routinely, to make sure that it's run. And also, again, there would have not been within the grid. We still apply like those reagents I was talking about that are control methods, and positive and negatives. And again, we wouldn't have had a match in the profile.
Wasn't there a possibility, though, that this instrument went into item 1, end of the cigarette, or I'm sorry, into my client's sample instead of the end of the cigarette butt sample?
No, once again, there's quality controls in the programming that we ensure. We have somebody that ensures that everything is programmed correctly into the machine before it's run. And that's what this ensures, is that they can match it up to that program and make sure that it's drawing from the correct tubes.
Your Honor, I have no more questions. Do you have any questions?
No further questions.